And with that, we conclude the awards part of the evening. And so it's now my pleasure to introduce this year's National Lecturer. Those of you who know me will know that I've spent most of my life working on ribosome structure and function, which is to say protein synthesis. And as a member of that fraternity, good standing, I can tell you that we've always taken the view that when it comes to the sort of downstream part of gene expression, that all the heavy lifting is done at the time you produce the polypeptide, because we were taught from childhood by Anfinson that proteins will take care of themselves once you get their sequences right. Our attitude, sort of cavalier attitude to what happens afterwards, is probably well captured by the lyric that Tom Lehrer once wrote about Wernher von Braun, the rocket scientist. And this is maybe some of you are a little too young to know this song, but the line I remember and that's appropriate here is the line that goes, once the rockets get up, who cares where they come down? That's not my department, says Wernher von Braun. Okay, well, Arndt Horwich cares where they come down. That's his business, and no one has made larger contributions to our understanding of how proteins actually fold in living organisms than Art. And this is protein folding not in the quiet world of the laboratory test tube at low concentration. This is the real world at high concentration in the macromolecular chaos, which is the cytoplasm of the average cell. Art got his start in education. He got both his bachelor's degree and his MD degree from Brown at the age of 24. And someday we're going to have a discussion about this, because it seems to me this means that he went to college at about the age of 12. But he'll explain that to me doubtless at some point. And after completing his MD at Brown, he did his internship and residency at the Yale Yale New Haven Hospital in pediatrics. And then he spent 3 years at Scripps as a postdoc, and then came back to Yale and spent 3 more years as a postdoc with Lee Rosenberg in the Department of Genetics. Well, Lee Rosenberg is a smart man, and Lee realized that he had something really unusual. And so Yale had the presence of mind to keep Art. Okay, Art is today professor of genetics in the medical school. He's HHMI Investigator. He's member of the National Academy, winner of numerous awards, amongst them the Rosenstiel Award and the Gärtner Prize. And so it's an enormous pleasure to me to present Art and turn the floor over to him. His title is Chaperonin-Mediated Protein Folding.
Thank you, President Moore. I am honored and humbled to be presenting this lecture. And thank you and the Society for inviting me. Thank you so much for your kind introduction. Mom, I wish you were here. You've been wondering for years what I've been up to. You keep asking when I'm going into medical practice. Here's what I've been up to. So, dear Society colleagues, what I want to present today is hardly the work of a single individual. It stands shoulder to shoulder with both scientists from my own group, but also with many other groups that have worked on these machines going back across decades. I fear that I can't do justice necessarily to everyone who's worked on these, but I'll try my best. And so my overall goal is to tell you a relatively short story, reflecting on how much fun it's been in my adult life to stumble on this remarkable machine that Mother Nature uses to assist protein folding in the cell, and to get to play with that machine on a daily basis for many years to see how it works. My talk will thus cover some of the history of the discoveries that were made, but I'll also seek to update concerning the latest understandings that we've gained. As a further comment, part of the great fun of this exploration has been the wonderful interactions with collaborators who brought and taught my group incisive biophysical approaches to understanding molecular mechanism. And I hope to convey how enjoyable that has been. So first, a little bit of history. Then my talk will proceed into structure and mechanism concerning polypeptide binding by chaperone machines, then polypeptide folding inside of them, a little bit on the ATP reaction cycle, and then a little bit of an overall movie at the very end. So the granddaddy experiment that I think we all pay tribute to regularly, that I heard about when I was an undergraduate at Brown in the early 70s when it was conferred a Nobel Prize, was the astonishing experiment of Christian Anfinsen. He basically took native ribonuclease with its 4 disulfide bonds, reduced it and unfolded it with mercaptoethanol and 8 molar urea, and took the essentially random coil protein, dialyzed it to remove the urea and mercaptoethanol, let it air oxidize, and it completely recovered its native active state. This was so astonishing to me when I heard about it. I was working in a fat cell lab as an undergraduate. We went and looked for every protein in the lab that we could find to try to reduplicate that kind of experiment. It was so amazing. The basic take-home, of course, from this experiment is that the amino acid sequence of a protein contains all of the information to enable it to fold into the unique native state, presumed to lie at the energetic minimum. So my life went on through medical training and so on and so forth, and it was nearly 20 years later that suddenly we reached a point where we actually could ask a question that related to this same area in which Anfinsen had worked. So through the work that we'd been doing with Rosenberg's group, we had been studying mitochondrial proteins that are encoded in the nucleus, translated in the cytosol as larger precursors with N-terminal signal peptides that direct them to mitochondria. And they then enter the organelles, in this case the innermost matrix compartment, and those signal peptides get cleaved by proteases, and the mature proteins were always assumed to spontaneously refold into their native state. I should point out that the notion that the proteins go through the membranes in an unfolded state was very clearly shown several years earlier to our work by Gottfried Schatz, who showed that, for example, dihydrofolate reductase with methotrexate bound to it and a signal peptide connected to it could not cross the mitochondrial membranes. But if you took away the methotrexate, the protein would now go right into the mitochondrial matrix via the leader peptide sequence. And so this suggested that proteins go through these membranes in an unfolded state. And the assumption, as I said, was always that they refold spontaneously on the other side. Well, we had been studying mitochondrial protein import in baker's yeast and had made a whole collective of temperature-sensitive lethal mutations. The idea being that if you can't make new mitochondria, you have a dead cell. That also was work of Jeff Schatz very early on that suggested that if you couldn't make new organelles, the cell would not survive. And so we decided that we could look in our bank of temperature-sensitive lethals for, in fact, a mutant where perhaps the precursor protein could enter the mitochondrial matrix, but it would require the action of some gene product or a machine or something of that sort in order to reach the native state. This was not so completely out of the box because workers like Hugh Pelham and others had been suggesting that there is a cohort of proteins inside cells called heat shock proteins that assist the disassembly under aggregation-prone conditions of other proteins, adjust their conformation in some sort of way. Here the idea was that maybe there could be something that actually assists productive folding to the native state under non-heat shock conditions in the complete absence of cellular stress. And no sooner did we have the idea to carry out such a test than within a couple days, Ming Cheng, my first graduate student, identified a mutant in our bank, a TS lethal mutant, in which indeed this particular marker protein, ornithine transcarbamylase, that we had been studying as a human urea cycle enzyme for years with Leon, but we're now using as a marker of mitochondrial import in Baker's yeast, failed to reach its native form inside the mitochondrial matrix. So in normal yeast cells, if you GAL-induce the OTC subunit, it goes with a signal peptide on it, it goes into the mitochondrial matrix. I should say yeast have no OTC in their mitochondria indigenously. It goes into the mitochondrial matrix, the signal peptide is cleaved, and the protein assembles as a homotrimeric active enzyme. And what Ming found was that there was no activity in this one particular mutant after temperature shift and induction of the OTC. The enzyme subunits indeed went, as far as we could tell, to the mitochondrial matrix because the signal peptide was cleaved, and yet there was no activity. There was a further test that we could do that directly assessed whether the OTC had trimerized, and indeed it showed that it had failed to trimerize. So it seemed like this protein had misfolded in mitochondria. And Ming began to study the endogenous protein, the beta subunit of the F1 ATPase, using a chloroform extraction procedure, and that also seemed to be failing to reach the native state. It didn't extract properly. And just at this time, we were saved in many respects in this project by a phone call from Walter Neupert and Ulrich Hartl. They were aware that we had been making some yeast mutants in the import pathway, and were interested in whether we needed any biochemical assistance. Well, of course we did. We were 3 people in a new lab who had little or no experience with yeast biochemistry and were sort of stumbling our way around. So they kindly invited me to come to Munich, and I explained to them mutants that we had that clearly affected the mitochondrial signal peptide processing enzyme. But at the same time, I explained to them this one remarkable mutant that we had called mitochondrial import function 4, or MIF4 for short, in which it seemed that proteins were being imported